So she got her PhD at Princeton, then went on to do a fellowship with the Rockefeller Foundation Planetary Health at Harvard, and now is at UC Berkeley since 2019. She does a range of work on things like RSV, chikungunya, measles, mumps, rubella, a lot of work in this disease demography and climate interactions. But also is like no stranger to controversial issues. And like I was looking at your adverse events with police officers and machine learning. That was an interesting one. And socioeconomics and COVID-19 burdens. Also, like just a ton of great work. So we're so happy to have you, Ayesha, and you can take it away. Thanks for coming.
Great. Thank you so much, Paul. Thank you for having me. This is, I feel like EEID is my favorite conference, and being able to have this other forum that's, happening year round is really, really great. Okay, I'm gonna share this worked before, so hopefully it works again. Is that good?
Looks great.
Looks
Okay.
great. Yeah.
Awesome. Okay, well, thank you so much. So I'm, as Paul mentioned, I'm a demographer by training, but I have really spent most of my career thinking about infectious disease dynamics, and I have, I think, spent most of my career independently thinking about the climate and population impact. So I do a lot of work on trying to estimate statistically, like what is the impact of climate variables on disease burden. And on the other side, I spent a lot of time thinking about, well, human behavior and population change. And recently, I've really been trying to think about these together and thinking about, well, what are some of these extrinsic and intrinsic drivers of infectious disease transmission? How do we, estimate these relationships? And then I also do a lot of work on mechanistic modeling. How do we take these statistical estimates and model, actually, put them into models to help us capture these nonlinear dynamics of infectious diseases? And then how do we think about how these changes might jointly or independently change the future burden of diseases? So this is very broad. And so today I'm going to talk very specifically about one particular aspect. And so I'm gonna focus today on flu, which is a pathogen that I've been interested in of late. And I'll show one study where I look specifically at climate impact. So there's many ways that climate impacts infectious diseases. I think this is most well studied, I would say, for vector-borne diseases. So I think Lisa spoke in the last time about dengue, I believe. So Lisa's work and work of lots of other people, including those on this call, like Aaron, I think have really highlighted how important it is to think about climate for vector-borne diseases. And there's been some more recent work, really led by Jeff Sherman and colleagues, thinking about how climate might affect the dynamics of respiratory pathogens like flu. And so I'll show some work on flu and climate, but I also think about demography. So things like, how does urbanization affect disease transmission by bringing people into contact in different ways than you would in a non-urban setting? Also thinking about population growth or decline. And in particular, I'll talk a little bit about aging. And aging is something that, in the demography world, this is really what a lot of demographers are spending a lot of their time studying these days. The sort of the rapid decline in birth rates that has led to very different age structures over a very short time span, and what this might mean for various health outcomes, various economic and social outcomes. And in today's talk, I'll talk a little bit about how we might think about aging populations and influenza burden in the future. So I think probably don't need to talk about this, but I think everyone in this call knows that there's various ways in which climate has been shown to affect infectious diseases. as I mentioned, the majority is probably work on this is probably on vector-borne diseases. Lots of really fantastic work there. Also on waterborne diseases, there's links between the famous papers on cholera, but also other waterborne diseases. And I'll focus on flu here, which is transmitted directly from person to person. There's airborne transmission, and some recent work has shown that well, actually, probably not so recent anymore, but work in the last 10 years or so has shown that things like humidity and temperature are correlated with seasonal peaks for influenza. And so I'll talk a little bit about some evidence that we find from Bangladesh. I should also say, just feel free to stop me and ask questions at any point, because this feels like a fairly informal group, so I'm happy to pause. Okay, so the first part is some work we did thinking about the impact of climate on influenza dynamics. And really, I think what one of the contributions we wanted to make with this paper is thinking about this in a tropical setting. So there's been, as I mentioned, studies that have looked at the link between climate and flu in temperate regions. So a lot of the studies that were carried out initially were in places where we had good influenza data, which happened to be temperate regions. But it turns out influenza in temperate regions can look quite different from influenza in tropical regions. So in temperate regions, the flu typically has the strong annual seasonal cycle, and we often see or almost always see the peaks during the winter season. So this is the kind of dry, cold winter season is typically when we see influenza peaks. And there's really, really fantastic evidence, both from lab studies, which is really key here. There's lab evidence for this to show us what mechanism would be behind this. But there's also evidence from more population-based and epidemiological studies that show that low humidity conditions in the winter can increase virus survival and enable the virus to transmit more efficiently between hosts. So Anis Lowen and colleagues did these fantastic guinea pig studies where they put guinea pigs in separate cages, and you can vary humidity conditions to see how transmission might differ. So these are some figures from a paper by Jeff Shemin and colleagues looking at the impact of specific humidity on virus transmission and then also virus survival. And you see this downward slope where you see higher or lower transmission at higher values of specific humidity, and you also see virus survival decrease as specific humidity increases. And then when we look at tropical and subtropical regions, the influenza season actually looks all over the place. So there's this really nice kind of not a review paper because it's not reviewing papers, but it's a review paper in the sense that it basically tries to look at what is the data that we have available, which we don't have a ton of data for tropical area, so that's an issue. But if we look at all the data that we do have for non-temperate locations, so subtropical and tropical areas, what we find is that some places you have transmission happening year-round. And you never really see a peak. You kind of see, ongoing transmission year-round. And then in some places you do see a peak, but the peaks don't happen in the winter like we do in temperate regions. But the peaks tend to occur typically during the summer months. But what actually, if you look closer at the data, what you see is that it's not necessarily the summer months, but for places that have a rainy season, we typically tend to see a peak during the rainy season. And so here's a nice figure from this paper where they look at specific humidity. So the left side of the paper looks very similar to what I just showed you from before, right? This downward slope. And here, instead on the y-axis, instead of looking at virus survival or data you would get from a lab study, this is just from population level flu case data. They're looking at what's the probability that there would be a peak. And so you see that the probability of influenza peaking declines as you increase specific humidity. And then you see this U shape where it goes back up again. And I will say we have a lot of evidence, specifically we have evidence from lab studies for a mechanistic reason for the left side of this graph. So there's mechanistic understanding of why you would see transmission go down as humidity decreases. I think we have a lot less evidence about what's going on the other side of the curve, why as humidity goes up again, as humidity continues to go up, you see this probability of seeing a peak go up again. And potentially this might mean that transmission is going up again as humidity goes up. So this is kind of the evidence that we have for tropical countries. And so we decided to have a look at data from Bangladesh. And one of the reasons we decided to look at Bangladesh is first, Bangladesh has a very clear seasonal pattern in influenza. And so influenza activity in Bangladesh peaks during the monsoon season, as has been seen in other tropical countries that experience a rainy season. But previous work has also shown that there's a spatial gradient in the timing of outbreaks across the country. And it's kind of, in it's paper by Berry et al. That was published in Lancet Global Health. They talk about various reasons of why the spatial gradient could be. So some of it could be human behavior. How connected are you to Dhaka City, which is right at the center there in the light blue? It could be due to climate factors. So they speculate about possible reasons for this spatial gradient. And so one of the things we wanted to do is, okay, well, can we explain the seasonality of influenza in Bangladesh by looking at climate drivers? And so do things like precipitation or temperature or specific humidity affect or allow us to predict these seasonal dynamics? Are we able to capture the peak timing and the latitudinal gradient? And then sort of a second order question that we wanted to ask is to think about, well, okay, if we do find that climate is implicated in disease transmission for flu, what might that mean for the future? Particularly because in places like Bangladesh, the climate change, so if you look at climate change models, making predictions about precipitation turns out to be really hard, and climate scientists have told me that this is it's challenging to predict exactly how monsoon seasons will change. But we do know that specific humidity is predicted to change pretty dramatically in Bangladesh. And so one might think that if there is a relationship between specific humidity and flu, then we might want to think about what the future burden might look like. And so we collaborated with some partners in Bangladesh to look at some influenza surveillance data. So Bangladesh doesn't have a sort of notifiable disease system. So there is no set system for the surveillance of infectious diseases. And a lot of the sort of reporting of cases kind of happens in an ad hoc manner. But one of the nice things is that they do have an influenza sentinel surveillance system. So they have hospitals across, I want to say they have 18 or maybe 19 hospitals across the country that will regularly do nasal swabs for patients that come in. They do it on the second Tuesday of every month. And they swab a certain number of patients who come in, and they then report this to the Ministry of Health, and the Ministry of Health compiles the data and makes this available. And so there's actually, this data is hard to work with because this is not a typical surveillance data, but there are signatures of things in this data that can help us get at questions of seasonality and peaks. And so what we have is 8 years of data from, we ended up using 12 locations because some of the newer hospitals were added so late that we really didn't have enough years of data. And so here in this plot on the x-axis is just time, and there's two y-axes. On one side, we're showing the total number of samples taken each month, and you can see that varies quite a bit. So that's the red kind of dotted line is the number of people tested where they did swabs. And then the green line is showing us how many people actually were positive for influenza. And so what we do is we convert this into the proportion positive, which is just shown by the blue curve, to account for the fact that different numbers are being sampled at different time points. And so in the subsequent results I'll show you, what we're really working with is this proportion positive. Now, we can talk about this is not a perfect measure for influenza burden in a country, but I do think it does capture sort of the seasonality and the peaks pretty well. And so, we try and we do a few different sensitivity things in the paper to try and address this issue. But in some ways, for most subtropical and tropical countries, this is the kind of data that we really have for flu in the absence of any sort of routine surveillance system. So that's kind of what a disclaimer and sort of what we're working with. So we took a look at sort of the seasonal pattern in the data, and we found a few things. So here in these plots, I am plotting temperature for every single plot. We're plotting temperature on the x-axis and specific humidity on the y-axis. And then we split it up by season. So we have a pre-monsoon season, which is actually the summer in Bangladesh. That's the hot season. And then we have the monsoon, the rainy season, which is roughly July and maybe June until August. So what you would think about summer in the US, that's the monsoon season. The summer season in Bangladesh happens before the monsoon season. And then we have the post-monsoon season, and then finally winter. And then each dot here is telling us it's a month location observation, and it's telling us the proportion positive for influenza. So the lighter colors is a lower proportion positive, and then the darker colors is a higher proportion positive. And you can see a few different things here. So influenza typically peaks during the monsoon season. You can see the really dark colors for monsoon. So peaks are typically in May, June, during the start of the monsoon season. And this is a time where you can see that the temperature is high and the humidity is the highest on average for the country during this time period. If you look at the monsoon figure, one of the interesting things is, and I'm sorry, you can't actually see it really well, you kind of have to squint, but there's some yellow dots that are to the right of the 30-degree temperature. So there's even just by looking at this, there's just seem to be what also previous studies have shown that at really high temperatures, you actually see a decrease in influenza activity. And so a lot of the monsoon activity is kind of concentrated right before you cross that temperature threshold. So there's a bunch of yellow dots that you can't see very well, but we'll talk a little bit more about temperature in a minute, but I wanted to point that out. Influenza burden can also be pretty high during the pre-monsoon season, but typically we only see this when specific humidity is also high. So those those purple darker spots are kind of concentrated in the pre-monsoon graph at the top corner, and that's when sort of you're at pretty high humidity level. In the post-monsoon, we see some activities and not a lot. And then in the winter, really, we don't see much influenza activity in Bangladesh. So it is a very seasonal dynamics in Bangladesh. In winter, you can see that the humidity is at the lowest and the temperature is at the lowest values as well. Okay, so what we did is we took this data and we first said, okay, well, let's try and fit some statistical models to try and see if weather variables are good predictors of the seasonality we see. And we tried various types of multivariate regression models. They all kind of showed similar results. So what we're showing here is the different predictors in the model. So we have on the x-axis the predictors, so specific humidity, temperature, precipitation. We have month fixed effects in the models, which means we're looking at variations within a month to estimate our effects. So we're looking at, for all the Junes in our data, how much of this variation can be explained by the specific humidity variation that we see within June. Okay, so that's kind of what those fixed effects are doing. And the fixed effects kind of confirm what we see is that you do see this kind of very seasonal dynamic where you have this clear monsoon peak and then maybe like a little peak in the post-monsoon season in the fall, and then really low activity during the winter months, which is January and February. So what do we find? We find this regardless of how we run the model and how we specify the parametric relationship or not, we've tried some non-parametric models of specific humidity and flu. We see this pretty clear U-shaped pattern which, was the Tamarius et al. Study also found for not quite in the same way. They were looking at the probability of a peak occurring, but very similar kind of shape, with the low point occurring roughly where the low point occurs for studies in temperate regions. So the left hand of the graph kind of looks like what you would see in a temperate region. So the predicted proportion positive for influenza goes down as specific humidity decreases, and then goes up again. And then for temperature, we have really wide confidence bands, and that's because the temperature range in Bangladesh really is very narrow. We don't have enough of a temperature variation when we put in month fixed effects. So within a month, there's not really much temperature variation that would allow us to kind of be able to predict what happens. But we kind of see this pattern that I was alluding to, where we see transmission go up, and then there seems to be some point after which transmission seems to go down. And this has also been shown in some of the lab work. So I wouldn't, obviously rely on our study to say that what the effect of temperature is, but because, Bangladesh is not the perfect place to study that. But the shape at least seems to be indicative. And then precipitation also is once you account for humidity and temperature. So these I'm plotting them separately, but they're all in the same model. So this is accounting for each other's. There's collinearities, which we can talk about how that might affect the results. But precipitation also, like once you account for it, it seems like specific humidity is capturing a lot of the variation that precipitation might have been capturing. So we find this interesting pattern, and we said, okay, well, there's some statistical association. What would be more convincing is if we took these statistical associations and put them in a mechanistic model, and then see if we can recover those patterns that we see in the data. So that's what we tried next. And what was really great for us is that Yuan et al. Wrote this really nice paper in PLOS Computational Biology in 2021, where they wrote up a flu model, but then they wrote up a nice way to characterize climate dependency. And so actually, anyone interested in this should go read this Yuan et al. Paper. I think it's really great. But they have a whole sort of category of ways in which you could characterize this climate relationship. And so we characterize it we borrow some of their methods to characterize R naught based on both humidity and temperature. So this figure is showing how R naught would vary in our model. And so we have this sort of typical U shape, or the tropical countries are thought to have. And then we have this temperature that also affects it. And so as you get into really high temperatures, you actually reduce your R naught. And then we estimate the exact shape of this. So we have three, no, that three is not right. We have 5 parameters, I believe, that govern this. Here I'm just kind of showing a general shape, but we have 5 parameters that govern this shape that we estimate when we fit to the data. And then the model we use is a pretty simple SIRS model. It's similar to flu models that have been used by Jeff Schraman and colleagues and others. But we basically have waning immunity. So you can get infected with flu, but after a certain period, you come back into the susceptible class. This is ignoring all strain dynamics and anything more complicated. It's really a pretty simple SIRS model. The only thing is here, our beta, our transmission term, has this climate dependency, and we're going to estimate when we fit to the data the exact parameters that's going to determine the shape of this dependency. And then we fit a few other parameters. I think we fix some things like the recovery rate and so on, but we do estimate waning, how long it takes before for waning immunity, and we estimate the initial proportion susceptible and a few other things. So we take this model and we try and fit it to our data. I won't really talk about the fitting methods, but we have lots of details on that in our paper, and I'm happy to talk about it if folks have questions. And we do I should say we do a few things. So one is we fit our model to the data to estimate our best fit parameters. And when we simulate, we leave out part of the time series. So we simulate up until we train the data and fit our model and get our best fit parameter estimates from using data from 2012 to 2018. And then we see how well when we simulate, the model does when we simulate forward. And then we do this for each location for which we have data. And so here the green is the observed data, and then the yellow is the simulated. And you can see that, I guess it really depends on what you think of as a good simulation or a bad simulation. I would say this is somewhere in the middle. We capture the seasonality pretty well. And the peaks we kind of can do. So if you look at the post 2018, so data was not we didn't use that data for fitting. We do okay, and we do better for some places than others. And it's sort of an open question as to what are the characteristics of those places and what else is going on that could explain the dynamics. We also, this is a pretty simple SIRS model, and as almost all of you probably have worked with these simple models and know that you can tweak these parameters to get these seasonal outbreaks. There's many ways in which you can get the seasonal outbreak. So in some ways it's not that convincing to be able to get the seasonal pattern. So we do exactly that. We try different kinds of models. So we have models where instead of having a climate dependence, we have sort of sinusoidal forcing that mimics something like school term time, which we know from past work that it's really important for disease transmission. And we also try models where we take out any sort of seasonal dependency. We have birth seasonality. We see how that does. And so we try out a few other models. The climate one does perform much better actually than the other ones, which we took as not like the final word on this, obviously, but it does seem to suggest that you kind of need this climate dependency to get the exact seasonal patterns that we see in the data. And then if we look more closely at seasonality itself, so the figure A is showing the monthly seasonality, and then you can see it peaking and then going down, and then our simulated data can do that pretty well. And this is really what we started with. Can we capture this latitudinal and longitudinal gradient that we see in the data? And a climate-driven model seems to do pretty well. You can see that the simulated lines have the same slope as the observed lines, but they are a little bit lower. And so our simulated incidence is actually a little bit lower, which could be, in the paper we talk about a few possible reasons this could be. I think one crucial thing that's missing from this, and what I do in my other work, is thinking about, well, these outbreaks often start in the big cities, and connectivity to cities probably matters a lot. We don't really have that in this model. We don't have things like what strain is circulating when, and what people's immune histories look like, and all of that, I think, probably matter. And then the other really big thing that probably matters is our observational model, and observational error is probably imperfectly capturing how this measurement process is happening. And so that probably is leading to it as well. Okay. And so we took this and we said, okay, well, what about, we have this mechanistic model now that we fit to current data. What if we look at what dynamics might look like in the future? So the figure on the left is looking at, so A and B are looking at temperature and humidity projections. So baseline is if we look at current conditions, and then projection is if you take these climate models. We use from the IPCC report, there's a whole suite of climate models, and we use the ensemble projections, and you can see that places, the green line shows that places are in Bangladesh on average will get more humid all throughout the year, but the largest jump is kind of during the monsoon season. And then we also see that there's a increase in temperature that's projected to happen on average across the country. And then we looked at what this might mean for each specific region. And what we found was interesting is that it really depends on where you are, kind of what your baseline humidity and temperature was, and where that location is going in terms of climate. So places like Dhaka doesn't really seem to change very much. Dhaka is the capital city. It's in the center. Whereas a place like Borisal, we see this shift in the timing, and then this like sort of flatter curve as well. And so, Borisal is right at the southern tip of Bangladesh versus some place like Sylhet, which is in the north west of the country. You can see that the outbreak is predicted to get much more sort of a more sharper peak. A lot of this has to do with exactly how humidity and temperature is changing. So in some places it gets too hot, and so the humidity effect and the temperature effect offset each other. So once it gets too hot, you kind of see decreased transmission, even if you're moving into more favorable humidity ranges. And then in some places, we kind of see the opposite effect. And so one of the takeaways from this is, there's not really a clear, sort of one size fits all projection that's works even for a country like Bangladesh, which geographically is a pretty, spatially it's a small country. And so even within a small country, there's a lot of variation in how climate might impact burden. So kind of just to summarize, we really tried to kind of figure out, how well does climate even predict what's happening currently? In general, we see that, our model can capture the temporal and spatial variation quite well. But I think it would be really interesting to think about this in the context of either a metapopulation model or to think about, something about connectivity, because I think that probably is having some influence on when peaks are happening in different places. When we project forward, we see, in general, a decrease in overall burden, and this is largely due to this temperature effect. So again, I think this is a really interesting and open area for research about, sort of mechanistically what's happening with the temperature effect, and also what does this temperature threshold look like beyond which transmission is reduced. And then in the winter months, the winter months actually move into the favorable humidity range in Bangladesh, or it's predicted to in the future move into this humidity range that you almost see sort of in the pre and post monsoon season. So the winter months are predicted to get a lot more humid. But this increase in specific humidity means that you actually may move into kind of the low. So because it's a U-shaped curve, you sort of move into this low transmission range. So so that kind of reduces the burden of flu. So lots of interesting kind of interactions happening. So that's kind of the first part, and I'm happy to take questions on this before I move on and talk a little bit about how we think about putting demography in here, if folks have questions. Or I'm happy to keep going. No, good. Okay. And I should say this is work I did with Rachel Baker and Pamela Martinez. We've collaborated a bunch over the years on thinking about climate variables and subsequent work. I'm going to show you some ways we've been thinking about bringing demography into this. So what we've been thinking about recently is, well, yes, these climate effects are important, but how do we also think about it in the context that demography globally is changing drastically as well? So there's a few ways in which demography affects infectious disease dynamics. I think, again, in this group, I think you all are very well versed in the ways in which demography is important. But the few things that I wanted to highlight, so things like birth rate, death rate, obviously important, but things that demographic drivers could themselves be impacted by climate. So one thing is, I've worked a lot on human mobility, and there's a lot of recent work, this really great paper in Nature Climate Change by Sui et al., looking at how climate might be affecting human mobility and displacement and where people are showing up. And so this map on the left is just showing a figure of how mobility has been changing drastically. So this is air travel routes going from 1933 to 2020, this massive expansion. And then the figures from Sui et al. Are really showing this disaster-induced changes in population migration and where people end up, which are likely to have consequences for infectious diseases. We might also think about urbanization and how urbanization and climate change might be interacting. I think this is sort of very relevant for vector-borne pathogens, for example, but also I think interesting to think about for respiratory pathogens. Ben Dale has a really nice science paper looking at the shape of flu outbreaks in cities versus non-cities in the US and what might be causing it. And it's probably some combination of demography and climate. And so I think that's another exciting avenue. And then finally, what I'll talk about is sort of population aging. So we've seen this very rapid decline in fertility rates globally. So this is the total fertility rate, which summarizes the total number of births an average woman would have in these countries. And you can see how this is changing over time. So this is going from 1950 to 2100. And you can see this sort of convergence of these fertility rates, right? So there used to be a lot of variation on the number of births per woman. But over time, as countries have gone through the demographic transition, you've seen this sort of convergence towards lower fertility, roughly converging around 2 children per woman, which is what in demography we call replacement level fertility, roughly 2.1, how many children you need to have in order for the population to stay stable. As we now know, in many countries, it's gone below 2. So countries in Asia, Japan, and Korea are sort of the classic examples, but also countries in Europe have dipped below replacement level fertility with fewer than 2 births per woman. And so these are countries that are projected to have a shrinking population going forward. So what does that mean? It means that the age pyramid is going to look pretty different in not a huge time span. So this is data from the US Census Bureau, and they had this really interesting infographic. They say for the first time in US history, older adults are projected to outnumber children by 2034, which is, kind of really like dramatic in terms of the scale of this. And the population pyramid is going to look pretty different, right? So we go from this sort of classic pyramid shape where you have more babies. And in 1960, you can see this sort of very classic shape. There's some effect of the world wars in the middle, sort of hollowing it out, but you can kind of see this classic shape. In 2060, there's 2 things that we think probably have the largest impact on disease dynamics. One is sort of the narrowing of the base, and so the birth rates are falling. And so, for childhood diseases, we want to think about what the susceptible population will look like. But also we have this widening of as baby boomers and the generation Gen X that came after baby boomers, as they age into those older age groups, we see both the size, so the number, because the population is still growing, our growth rate is positive, both the size of the population in the elderly group is going to increase, as well as their proportion of a population. And this, should probably even without doing any models, you would think would affect disease dynamics. I like to show this figure because I think it's a really brilliant piece of work looking at for a whole range of pathogens. I'm sorry, this is really small. Each figure is small, so it's hard to see, but this is for a whole range of pathogens. They're plotting the age dependency in case fatality rate. This is for 28 human infectious diseases. And this is work by Sorcy and Fev, published in PLOS Pathogens. And they show that, this age dependency, which we associate really for respiratory pathogens for flu and COVID, it's very clear. It's, even if you ask someone on the street, I think they would be able to tell you who is at risk. But for many other pathogens, we still see this clear pattern where the very youngest and the very oldest are often the ones most at risk. And so one of the things that I've been thinking about also with Rachel and then Kylie, who's a wonderful student with me here, is thinking about, well, how do we jointly affect the model of climate, jointly affect the impact of climate and demography? And Erin is editing a book chapter that we actually wrote this for. So thank you, Erin, for forcing us to sit down and write a model and think about it. It was really, really a good opportunity. And what we try to show is, if you take a climate-driven flu model, so here we have in some ways a very toy simulation, and this is for the US. I'm looking, again, pretty similar to the simulations I was showing before for Bangladesh, but now I've split it up by age group. So figure A is showing prevalence in 0 to 17. B is, I should have numbered this differently. C is 18 to 59, B is 60 plus, and then I'm showing the hospital burden. And the current is just showing if we take current climate conditions, we have a climate forced SIRS model. What does it look like? And then if we both project climate conditions and the aging structure of the population, so going from the orange age pyramid shown in figure E to the green age pyramid shown on the right, what does it look like? And there's a few interesting things. One is the climate conditions in the US are projected to change in a way that on average, overall burden should go down. And so as you move into the higher humidity, remember we're on that sort of left side of the U-shaped curve, we're moving into the higher humidity ranges where transmission is lower. But what happens is when you have an aging population, you offset this effect. So you can see that for the younger age groups, incidence goes down, but for the older age groups, prevalence actually goes up. And then we see this play out in hospitalization as well, where we see this hospital burden. The hospital burden looks a little bit different for 60 plus because some of the burden for hospitalization also comes from the under 0 to 1 age group. And so that age group sees a pretty dramatic decline because of predicted climate conditions, at least in our projections. Again, I think there's more work to be done in figuring out how carefully are we estimating these statistical relationships, how good are our climate models, and so on. But roughly, we see this offsetting effect. And this kind of led us to think about, well, there's probably this offsetting effect happening for other pathogens as well. And so one future direction is trying to think about, well, what will these falling birth rates and aging mean? And then just in 5 minutes, I wanted to say one other complication that I've really been thinking about lately in terms of flu is thinking about not just population aging, but our immune history and what population aging might mean. So this is a paper by my student, Kylie, that's about to come out. And what Kylie did is basically analyze a vast amount of mortality data for the US. And so the figure on the left is a heat map. And it's showing time on the x-axis, age on the y-axis. So this is a very classic like Lexis diagram. So the cohorts are going along the diagonal. These are birth cohorts. And what we're plotting here is excess mortality for influenza in red. And then the opposite of excess mortality will be called mortality deficit in purple. And what you can see that before the 2009 pandemic, we see a particular pattern where we have excess mortality in the higher age groups. And then after the 2009 H1N1 pandemic, we see a pretty different pattern. And the interesting thing we found was that we actually, see, and I should say these excess mortality measures are calculated accounting for age structure and all of that. So it's excess relative to what you would expect given the age profile of the population and so on. And what we see is that after the 2009 pandemic, the switch happens, but it actually only happens in the H1N1 years, which tells us, okay, the H1N1 pandemic in 2009, H1N1 years seem to be different, and other studies have shown this as well. And that difference actually seems to persist, and you can kind of follow the cohort lines. So for example, the 1940 or 1950 cohort lines, and you can see that the red bits are moving up every year as you would expect. And so we kind of ask the question, well, what's going on? What explains the fact that younger adults seem to be at higher risk during these H1N1 years? And other folks have hypothesized that this is part of this could be an imprinting effect. So what you're exposed to early in life, in the first 10 years of life, might have this long scale impact on what your severity of flu and mortality risk of flu looks like as you age and in the future. So the figure on the right comes from statistical models that we fit to the data to try and estimate this. And so the colors represent what the group or a birth cohort is imprinted with. We can go into how we estimate this. It's kind of complicated, but you can see like kind of what we would expect, that The older cohorts that experienced the 1918 flu pandemic, which was, antigenically, more closer to the 2009 pandemic than subsequent H1N1 outbreaks. These older cohorts really had pretty significant protection during the H1N1 years. And the H3N2 imprinted cohorts really seem to have sort of a larger burden than you would expect in the H1N1 years. What does this mean in terms of aging? Well, I think the kind of the troubling part of this is that these H3N2 cohorts are now aging into the high-risk age groups for flu. And so if we think about what the future might look like in terms of aging, I think there's probably some worry about, as these H3N2 cohorts that do worse in H1N1 years age into these higher risk age groups, and flu has a pretty strong age gradient, we might see a lot more hospitalization and severe cases. And so we're doing some work simulating what this might mean as the age structure changes and the cohorts that were imprinted with different things move into different age groups. So I think of this as sort of a further complication, but I do think this is a pretty important thing to consider in terms of population aging and flu dynamics. So I'm gonna just end with like a few concluding thoughts, which I think that there's already ways in which climate and demography are impacting disease burden and range. And I think folks in this series can probably speak to that much more than me. But there's lots of confounding factors, I think, and maybe in some work, when climate work is presented, demography is a confounding factor, but in some ways they're jointly affecting it. But there's many things I didn't talk about. There's socioeconomic factors, there's healthcare access, land use, different policies, human behaviors, thinking about how society adapts to climate change. All of these things are probably having an effect. And sort of, I think, thinking about ways to collect better data about these factors, not just in a way that typically I think there's lots of social scientists who work on these kinds of problems, but really we kind of need this data collected in a way that we can then statistically estimate those relationships and then put them into these models so we can think about the mechanistic structure and the nonlinear dynamics. So I think thinking about how to cleverly collect these kinds of data for modeling purposes, I think is really going to be important. And then lastly, I think the impact is not going to be equally felt by all groups in the population. I think the aging population in particular, we care about for respiratory diseases, but also other pathogens, but then also thinking about minority groups and those in low-income communities and how burden might differ across groups. So I'm gonna end there. A lot of this work was with Rachel Baker, who's at Brown. So I just want to point that out. And then also Kylie, who's my PhD student, did a lot of this amazing work. And I have my funding sources there. And thank you. Happy to take questions.
Thank you. Any questions? Well, while I'm waiting, I have questions. Oh yeah, okay, go ahead. Somebody had a question? Okay, I'll go. So Ayesha, what do you think the mechanism is for the increasing part of the U curve on the humidity sort of thing? Like what's going on there? My understanding was you have data for the one side, but you just have a hypothesis on the other side. So what do you think's happening over there?
You're right. I can share again if we need to, but that's a good question. I am really, I would say, not the right person probably to answer that question. I think that it would be great to have some lab studies, and there might be lab studies ongoing already that I don't know about that look at that. But I think that the original lab studies were trying to confirm what we saw in the population level data for temperate countries, and so it was kind of restricted to the ranges that we saw. I don't really honestly know enough about the biology of influenza viruses to dare hypothesize about what it could be, but I do think what we need is probably maybe some lab studies. But maybe folks on this call have ideas of what is going on.
Marm, you had
Yeah,
a question?
seems
Oh,
like a
sorry.
hole in what we're measuring. Like there's like things that we should be measuring that we're not in the lab.
Marm, do you
Sorry,
want to go?
yeah. Marm and
Okay,
then Jamie. Yeah.
great. Thanks very much for a really nice talk, Aisha. I was curious, I might have missed something, but it seemed like when you showed us the data for Bangladesh and the kind of range of humidities that were in Bangladesh and then the kind of fits afterwards, it seemed like the left-hand side of the U shape for humidity was in a range which I didn't think you actually had any data in Bangladesh at all. So I was curious if you were borrowing the temperate data stuff from that to get the left side and then fitting mostly the right side, or how those two things kind of coincided.
Yeah, I wonder if I can just share so people know. Thank you, Marm. That's a really great question. Okay, let me find the
That
slide.
one, yeah. So like
Yeah,
the bottom of your graph starts at 8, right? And then I think on the next slide, all of the decrease is happening in the like 0 to 5 range. So I was curious. Yeah, that part
yeah,
exactly.
yeah. So we did a few things. Um when we fit this, we had tried pulling in some, kind of pooling data from other countries to try and fit this. Um, and you can see that the confidence bounds are much wider. I don't know if you can see my
Yep.
my screen. So the confidence bounds are, much wider. And then when we actually estimate the relationship, we actually don't even see this. I'm showing this here like schematically, but when we estimate the parameters to fit the data, we don't really see this. So we kind of just get this part in Bangladesh. So that, yeah, it's
Yeah,
it's
cool.
it's
So
like that
basically,
other side is more relevant
it's
for
coming from the
Bangladesh.
other temperate regions. Got it.
Yeah,
Cool. Thank you
yeah,
so much.
yeah.
Um, I'll jump in. Great talk. Thank you very much. A couple of comments. One is on the humidity point. There is lab data that shows increasing half-life of viruses as relative humidity increases. And that didn't seem to show up in the absolute humidity data that you showed from Jeff Shaman's lab. But there's a paper we wrote with virologists that shows it's a U-shaped response in terms of half-life. I wouldn't dare postulate that that drives this macro transmission effect that you're seeing, but it's a factor that could play a role. My question was about the cool imprinting stuff you showed at the end, and how, it makes sense, I guess, that within H1, because it's the same subtype, you're probably getting like an immunodominant response to the sites in the head, the binding pocket, and all that. And so you might expect that to show more the sort of drift uh, evolutionary response that you see within subtypes typically. I'm curious whether there's enough data, I just don't know this, about like antigenic distance to, provide a kind of molecular scale correlate of why the particular years look like a few decades worth of H1 provided protection while the later H1 didn't.
Yeah, that's such a good question, Jamie. Thank you. And also for your first point, Jamie. Thank you so much. Um, Kylie spent a really long time trying to figure this out. Because that it did seem, especially maybe I could share my screen one more time. So if we look at, it did seem very cohort specific in some ways. Like exactly which cohort saw which version. And so in the paper, we talk about 3 different eras of the 1918 H1N1. We talk about an alpha and a beta and a gamma, and that is from some past work looking at this question of how similar is the 2009 H1N1 pandemic to those strains. It does seem to be at least from what we could gather from the literature, that the H1N1 alpha, which is like the earliest one, does look most antigenically similar to the H1N1 2009 pandemic. But I would say that, we kind of, again, we sort of say this and our data, we don't have good statistical evidence to like really differentiate like exactly which cohort, because that's hard it's hard to do the sort of age period cohort. It's really hard. And so unfortunately, I mean, there's probably you kind of, I think this mortality data is really great because we have single year of age, we have every single year in the US. But it's limited in that ability to really disentangle and then the trouble it's the other trouble is sort of assigning imprinting probabilities to cohort properly, and I don't know if we do a great job in the paper, but we attempt. Yeah. Thank you.
Are there any other questions from the group? I have one if not, but don't want to take up time. So Aisha, I wanted to ask about the first result that you were showing with the U-shaped patterns, and then also like some of the discrepancies that you talked about just geographically. And you talked about connectivity to the main capital as potentially like one of the reasons for those discrepancies. But I was also wondering, kind of related to the U-shaped pattern, is there any difference in human behavior throughout the monsoon season? Like you talked about accounting for school seasons, but maybe is there some, internal air conditioning or, shelter that people are taking during these seasons that's facilitating behavioral drivers of transmission? And then kind of getting to that idea of like local or geographic discrepancies. Is there discrepancy in who actually has air conditioning geographically and potentially like, the role of congregating in indoor spaces in disease transmission?
Yeah, that's such a great question. Well, there is a really fantastic paper by Shweta Bansal and her group looking at exactly this question, because some of this climate dependence can be, like you said, human behavioral, where people aggregate and who has access to air conditioning and who doesn't. I think Shweta's work is in the US, so her looking at sort of seasonal variation in this. And then I think Shweta might even have a separate paper on the air conditioning piece. I've definitely seen something from her group. But their group has thought a lot about like how much is it this human behavioral aspect versus this climate aspect. In Bangladesh, I mean, it's interesting. I mean, most of the population doesn't have air conditioning except some folks in the capital. But I think that where you aggregate and how you aggregate probably does vary a lot depending on whether it's the monsoon season or the winter season. The winter season is in Bangladesh, it's interesting. The winter season is the season for it's like wedding season. So it's like massive events, and these are like very large scale events with like thousands of people, like villages will have weddings. And so there's mass gathering of a different form versus in the monsoon, maybe you spend more time indoors and there's, so it's hard. I think it would be interesting to link it. In my other, like when I wear my other like more demography hat, I do a lot of work on measuring like contact patterns and human mobility. And it would be interesting to like kind of replicate some of Shweta's work in Bangladesh, looking over time seasonally, how our behavior contact changing. It's a really great question.
Awesome. Thank you. Any other questions from the group?
Maybe let's thank Aisha for coming. Thank you.
Thanks so much.
Yeah, we'll cut off the recording.